In this video, I'm going to talk about DNA transcription. The key concept to remember when we talk about the pathway of DNA to protein is what is known in molecular biology as the central dogma. And that's just the fundamental principle that DNA is transcribed into RNA, and then RNA is translated into protein. And we use the term transcription for the generation of ribonucleic acid, or RNA, from DNA because it emphasizes the fact that DNA and RNA both use the same general language of nucleic acids. I sometimes think of monks transcribing ancient scrolls when I think of DNA transcription, because when they were transcribing the scrolls, they were simply making copies of the text in the same language. Also remember that DNA is the template for RNA. And the synthesis of proteins according to the genetic code that's carried in a molecule of RNA is that second step in processing the genetic information. So remember that proteins provide structure, and they also carry out many essential activities in the cell, and also that transcription takes place in the nucleus of a eukaryotic cell, but translation takes place in the cytoplasm of the cell. The term translation is used for taking RNA into protein because it suggests a change in the language that's used, and that change is from nucleic acids to amino acids. It's like translating English into Arabic. Nucleic acids are the building blocks of DNA and RNA, and amino acids are the building blocks of polypeptides and proteins, in a similar way that English and Arabic have different symbols or alphabets that create words in each language. Proteins are synthesized on ribosomes that are present in the cytoplasm of a cell. But chromosome DNA is found in the nucleus, so to get the message from the nucleus to the ribosomes, there needs to be an intermediary molecule to do that. That intermediary is ribonucleic acid, or RNA. So RNA and DNA are both nucleic acids, but they have a bit of a different structure. For example, RNA has ribose as its sugar, whereas DNA has deoxyribose as its sugar. So deoxyribose is named because it's missing an oxygen atom on the 2' carbon. So in a sense it has been deoxygenated. Another difference between RNA and DNA is that RNA has Uuracil, which ends up replacing the base thymine that is found in DNA. Although the structure is similar, Uuracil has a hydrogen where thymine has a methyl group. So this hydrogen in Uuracil is actually a methyl group in thymine. To summarize the differences between DNA, remember that DNA has a deoxyribose sugar, whereas RNA has a ribose sugar. The bases in DNA are adenine, thymine, cytosine, and guanine, whereas in RNA, Uuracil replaces thymine. The 5' end of DNA is a monophosphate, but the 5' end of RNA is actually a triphosphate. DNA is relatively large, whereas RNA is smaller. And finally, DNA is double-stranded, which makes it really stable, but RNA is single-stranded, which is part of the reason that it's more prone to degradation than DNA. The general process of transcription is straightforward, and it's similar to DNA replication in many ways. As a region of the DNA unwinds, one strand is used as the template strand. And that template is used as a guiding system for the RNA transcript which is generated. The other strand is the non-template strand. But RNA uses Uuracil instead of thymine, so the A's in the template strand are matched with U's in the RNA transcript instead of T's. The enzyme responsible for this process is RNA polymerase. And the new strand grows in the 5' to 3' direction, which means that the template DNA strand is in the 3' to 5' direction. The initiation and termination of transcription involves promoters and terminators. So RNA polymerase and associated proteins bind to the DNA duplex at the promoter region. Eukaryotic and archaeal promoters contain this sequence which is similar to this TATAAA, and that's known as the Tata box. The first nucleotide to be transcribed is usually positioned about 25 base pairs from the Tata box. The strand that gets transcribed differs depending on which gene is being transcribed. So the template strand could be either strand depending on which gene is being transcribed at the time. An RNA polymerase will move along the template strand in the 3' to 5' direction because remember it builds in the 5' to 3' direction. And transcription will continue until the RNA polymerase encounters this terminator sequence. And transcription is regulated, so genes that are needed all the time in all the cells, often referred to as housekeeping genes, are transcribed all of the time. But most genes are only transcribed at certain times and in certain conditions or in certain cell types. So there's a process in which the promoter region in eukaryotic DNA is recognized to start the process of transcription. There are enhancer sequences and a promoter region that has the start site of transcription. In eukaryotes, transcription initiation requires the combined action of at least 6 different proteins called general transcription factors. General transcription factors bind to the promoter region and activator proteins bind to the enhancer sequence. The next step in promoter recognition is that the transcription factors recruit RNA polymerase II to the transcriptional start site, and that's located in the promoter region. In addition, the transcriptional activator proteins that are bound to the enhancer sequence recruit a mediator complex, which then interacts with the RNA polymerase complex. So overall, these proteins need to come together and loop the DNA such that transcription can be initiated. Transcription takes place in what looks like a bubble that is about 14 base pairs in length, and the RNA-DNA duplex in the bubble is about 8 base pairs in length. The RNA polymerase II adds the nucleotides to the 3' end of the RNA transcript. The RNA transcript is extended through a polymerization reaction. So here we have the template strand and the RNA transcript, which is growing in the 5' to 3' direction. The incoming ribonucleoside triphosphate is recognized by the RNA polymerase and joined to the growing transcript if it base pairs correctly. So in this case, the A in the DNA strand would need to be paired with the U in the transcript. The RNA polymerase orients the oxygen in the hydroxyl group of the 3' end on the growing strand into a position from which it can attack the innermost phosphate of the triphosphate. The 3' hydroxyl of the growing strand attacks the high-energy phosphate of the incoming ribonucleotide, providing the energy for the reaction to take place. A pyrophosphate group is then released by a different enzyme, and this process makes the attachment of the ribonucleotide permanent and allows the next one to come in. In prokaryotes, the link between transcription and translation is simplified because the primary transcript is the messenger RNA that gets translated. So it's possible for both the transcription and translation to take place in the cytoplasm of a prokaryote. But in eukaryotes, there's an issue of the nuclear membrane that's a barrier to transport from the nucleus, where transcription takes place. to the cytoplasm, where translation takes place. So the transcript needs to pass through the nuclear membrane to the cytoplasm, where then the primary transcript and the mRNA can be translated into proteins on the ribosomes. Before the primary transcript can be translated, it needs to go through a number of different modifications. The first of these modifications is the addition of a 5' cap. Without the addition of the 5' cap, the ribosome would not recognize the messenger RNA, and then translation wouldn't occur. So the cap consists of adding this 7-methylguanosine to the 5' end of the primary transcript, and then this modified nucleotide is linked by its 5' carbon to the 5' end of the primary transcript by a bridge that's composed of 3 phosphates. The second major modification that occurs is the addition of about 250 consecutive adenines to the 3' end of the messenger RNA, and this is called the poly-A tail. So the process is known as polyadenylation because there's a lot of adenines being added, so poly means many. And this modification plays an important role in transcription termination, as well as the export of the messenger RNA to the cytoplasm of the cell. Both the 5' cap and the poly-A tail help to stabilize the RNA transcript because single-stranded nucleic acids can be unstable and susceptible to breakdown by enzymes. The third modification of the primary transcript is the excision of certain sequences known as introns, and these are typically regions that don't code for proteins, but they leave the exons intact, and they need to be connected together after the introns are excised, and the whole process is known as RNA splicing. So in the first step, specific sequences near the ends of the intron undergo base pairing with RNA molecules in the spliceosome and are brought into close proximity. The spliceosome enables a reaction that cuts one end of the intron and connects it to a nucleotide near the other, and it forms this characteristic loop called a lariat. The exon on one end of the intron is brought close to the exon at the other end, and then the exons are joined and the introns are released and the lariat breaks down. About 90% of all human genes contain at least one intron, and many genes contain many introns. In alternative splicing, primary transcripts from the same gene can be spliced in different ways to yield different messenger RNAs and ultimately different protein products. So if the introns are removed, but the transcript gets spliced at the end of exon 2 and at the beginning of exon 4, we end up with an mRNA that has a protein coding for exons 1, 2, and 4. Alternatively, if all the introns as well as exon 4 get removed, such that the protein coding remains for exon 1, 2, and 3, we create a different protein because exon 4 isn't on the end of the mRNA. So at this point, the messenger RNA is ready to be translated into protein by the ribosomes in the cytoplasm.